Today I'm going to go over 4 biological molecules that we'll be covering this unit: carbohydrates, lipids, proteins, and nucleic acids. Before I get started, I just want to mention just a couple of things that we're going to be going over. So we're going to be talking about the main function of each of our polypeptides. We're going to be talking about the monomer, or the building block. So monomer is another way of saying building block of our molecules, as well as what that structure looks like. We're going to talk about polymers, which are the larger molecules that are made up of the building blocks and what they look like. And then we're going to talk about common examples. So just to reiterate, monomers are our building blocks. You can think of them as the Lego pieces to whatever you're building. So each of the blocks that you need to put together in order to make your larger molecules. And polymers are your larger molecules that you made from your building blocks. Okay. So we're going to start with carbohydrates. Now, carbohydrates' main function, as you guys saw in the video, is for energy storage. So most of our energy comes from carbohydrates. But in some cases, it can also help and be a structural component. And I'll talk about that briefly as one of the examples. But for its main job in most cells is energy storage. So the building blocks of carbohydrates are monosaccharides. And so mono means one, and saccharides are what we call sugars. So it's basically a single sugar molecule. So one, and saccharide is sugar molecule. So this typically is pretty small. A really good example of this is glucose. So this here is a picture of glucose. You don't have to be able to draw this picture, but just be familiar with what a structure of a monosaccharide looks like. And so they all typically look kind of like this glucose molecule. Now, once we have a single saccharide, they can start to bond together and form a polymer. Or in our case, because we're talking about carbohydrates, it forms what is called a polysaccharide, which just means many sugar molecules. So you'll see here that I've basically just have a whole bunch of glucose in this case, all bonded together right here at these oxygens. And so these long chains that we form are called polysaccharides. And you can have lots of different polysaccharides. So starches are a polysaccharide, cellulose is a polysaccharide, and these chains can be tens of saccharides long, they can be hundreds of saccharides long, they can be thousands of saccharides long. So what are some common examples of carbohydrates? Well, we've got our very simple sugars. These tend to be our disaccharides and our monosaccharides. So monosaccharide being like glucose. Disaccharides are just 2 saccharides bonded together. A really good example of this is lactose, which is the sugar that you find in milk. And then we've got more complex carbohydrates like starches. And starches are going to be your polysaccharides because they're very, very long chains of sugar molecules. You also have something called cellulose, which is an interesting polysaccharide because although it does store energy, most of us can't digest cellulose because cellulose is found in lots of plant products like grass and leaves and stuff. Cellulose not only acts as energy storage for some organisms, the ones that can digest it, but cellulose is a really important structural component of the cell walls to give them more integrity. All right, so that's a little bit about carbohydrates. Our next biological molecule that we're going to talk about are lipids. So lipids also do some long-term energy storage. So once you've depleted all of your carbohydrates or a lot of that easy energy, your body will start to utilize fats or lipids of your body in order to also get energy. They also are an integral part of our cell membrane, and I'll talk about that in a little while. And they also form the base structure of some of the hormones in our body. So, those are three of the main functions of lipids that we're going to chat about. But before that, lipids are a little bit different than our other biological molecules because there are actually two types of monomers that we find in our lipids. So one of the monomers is glycerol, and glycerol is this part right here that I have boxed in blue. The other monomer are the fatty acid chains. So each chain is a monomer. So fats or lipids tend to be made up of glycerol as well as fatty acid chains. And we can talk more about this later, but if we're looking at our fatty acid chains, you'll notice that this one here up at the top, the one that I have labeled saturated, has a bunch of carbons that are all bonded together, but you'll notice that there's tons of hydrogens all over them, and it makes sort of a straight line. Saturated fatty acid chains tend to be the ones that get labeled bad, and that's because these saturated ones can lay flat, and because they can lay flat, those molecules lay closer together and are more likely to form a solid, which inside of your arteries and inside your body can be bad. Versus unsaturated, which I have pictured down here, you'll notice that there is a double bond here, which causes a kink in the shape. And that kink makes it so that these chains can't lay flat. And because they can't lay flat, they're more likely to stay in a liquid phase at room temperature, which makes them considered more of the healthy type of fat. Now, when we are looking at the polymers, so the monomers are glycerols and fatty acid chains. When we're looking at the polymers that form, we have to put them together. So I've already actually put them together in this picture, but you can put them together in other ways. So the most popular diagram you'll see are what are called triglycerides, are your fats. That's what I have pictured to the left. So you'll see the glycerol group, and then you'll see the 3 fatty acid chains. So those 3 fatty acid chains is where the tri and triglyceride comes from. And that's typically what we're talking about when we say fats, like fats in your diet or fat in animal fat. And if I were to draw a less complicated version of that picture, it would look more like this guy up here. So the glycerol is this round circle, and each of these legs is one of the fatty acid chains. Another way we could put together these monomers is the phospholipid, so the one boxed in orange. So here I see my glycerol. Instead, I have only 2 fatty acid chains, but on top of that, I've added additional group. That's just a phosphate group up there, and we'll talk more about what that is important for when we get to the cells. But when I draw a phospholipid, if I wanted to draw it less complicated, it would look more like this guy down here, where the glycerol is that circle on top, and then the 2 fatty acid chains are those 2 legs coming out the bottom. And because of their special properties, when these molecules come near each other, they like to form this sort of bilayer. Again, we'll talk more about that when we get to the cell membrane. The last one are steroids. They don't look anything like any of the other ones. There is no glycerol group. There are no fatty acid chains, but this is still considered a lipid. And it is these little hexagon, pentagon type boxes that are all together. So typically there are 4 of them that are all linked in a lipid. And that is just another structure. So I just don't want you to be thrown off if you ever see a structure like that and it's labeled as a lipid. So what are some examples of these? So again, really popular example are triglycerides, which are technically the fats, and those can be unsaturated or saturated. Our cell membrane or our phospholipid bilayer. Phospholipid is another example of where we see our lipids. And the other one are steroids. An example of this would be cholesterol. So those are some examples of our lipids. We're going to move on to proteins next. Proteins make up all kinds of different functions of the body. So I'm just going to label some of the jobs that we know that they do. So we've got transport proteins, we've got messenger proteins. They do so many jobs in the body. It's hard to list all of them here. We've got enzymes in the body. They act as structural components for cells. We've got, I already wrote messenger proteins. We've got storage proteins. So that's just a few of their functions inside of the body and what they do for cells. Now for proteins, all proteins are made up of things called amino acids. So amino acids are the building blocks of proteins. And every amino acid has this general structure, the one I have boxed in green. So nitrogens bonded to carbons, bonded to oxygens, they all have that general structure. Now, in the human body, there are about 20 different amino acids. So what makes one amino acid different from another amino acid is this very special group right here called the R group. So each of your 20 different amino acids has a different R group. And we'll look more at that later on when we get into genetics. But in order to make this easier, down here, instead, I've just made colored dots. And we're going to imagine that each colored dot is a different type of amino acid, meaning that each one has a different R group. So all proteins are made up of these amino acids. Now, before it's a protein, they have to first attach to one another. So the first step is to form what we call a polypeptide, or a chain of amino acids. So a polypeptide is just a chain of amino acids. So I have these amino acids and they're bonded together and they form just a linked chain. A polypeptide by itself really can't do anything yet, but it is an important first step to making our protein. After our amino acids are linked together, what happens is that they then start to fold. So some of them are attracted to each other, some of them want to be near other ones, and what happens is they start to fold into a pattern. And once our proteins fold into a certain pattern, then they will have a function. So what we often say is that a protein's shape will then dictate its function. Oops, hold on. So not until the protein folds into an appropriate shape will it function the way that it's supposed to. So what are some examples of proteins? Well, we've got transport proteins like hemoglobin. So hemoglobin is a protein in your blood cells that helps transport oxygen. We've got important proteins in your immune system called antibodies. So we've got enzymes in your body like lactase, which we'll be using later this week. So that's an enzyme, and its job is to break down lactose, and we'll talk again more about that, I think, later this week. We've got special receptor proteins that are on the outside of cells. We've got structural proteins like keratin, so that's an example. Keratin is in your hair and your nails and gives structure to those. So that's a little bit about proteins and their shape and structure and their building blocks. The last biological molecule we're going to talk about are nucleic acids. And nucleic acids, their job is to store genetic information. And we'll definitely be talking a lot about nucleic acids later on when we get to genetics. There's a whole unit on it. But just a quick overview. All nucleic acids are made up of monomers or building blocks called nucleotides. And nucleotides are actually also made up of 3 parts. So all nucleic acids are made up of nucleotides, and nucleotides are also made up of 3 things. The first thing is a phosphate group. So all nucleotides have a phosphate group, they have a sugar, and they have a nitrogenous base. And for those of you that are already familiar with nucleic acid, there are actually 4 nitrogenous bases, 5 if you know RNA, but let's stick with 4 right now. We've got adenine, which is the A, thymine, which is the T, cytosine, which is the C, and guanine is the G. So our nucleotides are made up of 3 parts, phosphate group, sugar, and a nitrogenous base. And there are 4 different nitrogenous bases that exist: adenine, thymine, cytosine, and guanine. So that basically means that there are 4 types of nucleotides. So all big nucleic acid molecules are made up of nucleotides. So I've got 2 examples, the ones that we're going to be working with the most, DNA and RNA. So DNA, you can see if we look closely, it's just made up of a whole lot of nucleotides that are all bonded together. So here is a nucleotide, here's a nucleotide, here's a nucleotide, and on and on and on. So it's just nucleotide bonded to nucleotide bonded to nucleotide, because those are the building blocks of a DNA molecule. And for us, DNA molecules are hundreds and thousands of nucleotides long. They're incredibly large molecules. RNA is another polymer or another big molecule that is made up of nucleotides. You'll notice that in this case, there's only one side, but they're still made up of these nucleotides. And you'll notice I have a U in here. So for those of you that already know, that stands for uracil. But if you don't know, we'll come back to that later on when we get to genetics. But it's still nucleotide after nucleotide all bonded together. So these are our 2 examples of nucleic acids. So DNA and RNA. And we'll be studying those specifically when we get to genetics.